Look at this sad little dog. Look at this clear blue pool that's usually filled with kids swimming in it. You know why? All because of one stupid little particle that we've all come to hate. What is it called, you may ask? Well, the virus. In this video, we'll be talking about what makes up a virus and how it works. So without further ado, let's get into it.
Until you find your way back home.
A virus is defined as an infectious particle consisting of little more than genes packed into a protein coat. Viral genomes defy the conventional thinking that genes are made up of double-stranded DNA, as we've talked about in previous videos. These genomes in viruses can consist of double-stranded DNA, single-stranded DNA, double-stranded RNA, which is ribonucleic acid, or single-stranded RNA, depending on the type of virus, which also constitutes if a virus is declared as a DNA virus or an RNA virus. One thing to note, the difference between DNA and RNA are the nucleotides. RNA has uracil instead of thymine. So as compared to the twisting and turning that DNA goes through to form chromosomes, in eukaryotic cells, the viral genome is usually organized in a single linear or circular way. Additionally, on average, viruses contain considerably less genes, only about 200 to a few thousand in their genome, much less than the estimated 20 to 25,000 in a human cell. Enough talking about what's inside the virus. Let's talk more about its pesky coat surrounding it. You'll see why it's pesky in a little bit. The protein coat surrounding the viral genome is called a capsid and can be either rod-shaped, polyhedral, or more complex in shape. Capsids are made up of a large number of protein subunits called capsomeres, with very little variety in the actual types of proteins present within them. So it's usually just one type of protein. Some viruses have accessory structures that help them infect their hosts. For example, viruses found in animals usually have viral envelopes surrounding their capsids, which are derived from the membrane of the host cell and contain host cell phospholipids and membrane proteins, making it easier for these viruses to engage with human cells because their viral envelopes kind of imitate the cellular membrane, and the cellular membrane starts to recognize it instead of, not recognizing it. Another complex capsid structure is found within a group of viruses that infect bacteria called bacteriophages, which in my opinion honestly are the coolest looking viruses because they just look like tiny robots, but they definitely have a lot going on. The capsids of these viruses have elongated icosahedral, long word which means a polyhedron with 20 faces, heads, and attached to the head is a protein tailpiece with fibers by which phages attach to a bacteria. Now, how do these viruses actually replicate if they don't have the equipment to do so? As we said before, they only really have a genome and then a protein surrounding it. Well, viruses can only replicate within a host cell, which can only be from the cells of a host species specific to that virus, which is one of the reasons why some viruses are specific to humans. These replications begin with the virus identifying host cells by a lock and key fit between viral surface proteins and specific receptor molecules on the outside of cells. Then these viruses bind to the host cell and release the viral genome into the host. The way the viral genome enters the cell depends on the type of virus and the type of host cell. For example, bacteriophages use their elaborate tail apparatus to inject DNA into a bacterium, while other viruses are taken up by endocytosis, which is kind of like when the membrane eats the virus and engulfs it, by fusion of the viral envelope with the host's plasma membrane, as I said. Once the genome is inside the cell, the proteins it encodes takes over the cell, reprogramming it to copy the viral genome and manufacture viral proteins. This slowly but surely takes over all of the cells. Oh wait, you hear that? It's time for some science history. the discovery of the virus with Moz.
Hey guys, Moz here. The discovery of viruses began with the study of the tobacco mosaic disease, a disease that stunts the growth of tobacco plants caused by the tobacco mosaic virus. In 1883, a German scientist by the name of Adolf Meyer discovered that he could transmit the disease from plant to plant by rubbing the sap of an infected plant on another. But when Meyer studied the sap under a microscope, he could not find an infectious microbe. This led him to believe that the disease was caused by an unusually small bacteria. This hypothesis was tested a decade later by Dmitry Ivanovsky, a Russian biologist who passed infectious sap through a special filter meant to remove bacteria. After filtration, he found out that the sap still produced mosaic disease, again reasoning that the bacteria was small enough to pass through the filter. It wasn't until a Dutch botanist named Martinius Bijornik, I don't know if I pronounced that right, sorry, did the discovery that it wasn't bacteria, but a particle much smaller and simpler was causing this disease to occur. Now passing it back over to Kabir.
Hey guys, y'all probably guessed, but the small particles Moz was talking about were indeed viruses. Anyways, one thing we hadn't mentioned was the structure of the virus we have all come to know, the coronavirus. Whoa. Coronaviruses have extraordinarily large single-stranded RNA genomes, approximately 25,000 to 32,000 bases, or RNA letters in length, and are surrounded by an envelope, which usually have a crown or corona of club-shaped spikes on their surface. So that's like an incredibly large genome. As we said before, most viruses only have like 200. Well, that's it for today's video. Thanks for watching the video, and make sure to like, subscribe, and comment what y'all want to see in the next one. Have a biotastic day.